When a virus replicates, mutation in its genetic sequence occurs, which may alter some characteristics of the virus. If the altered virus mutated significantly enough from the original virus, and gained survival advantages through these mutations, it may spread rapidly in a host population, becoming a new variant. The SARS-CoV-2 Omicron variant is a mutated strain from the original SARS-CoV-2. It includes the B11529, BA2 and BA3 strains. The Omicron variant was first found in September 2021 in South Africa, but upon further genetic sequencing, it is believed that this variant originated and circulated from elsewhere. As of February 2022, it has spread to at least 138 countries with a cumulative prevalence worldwide of 14%. While in South Africa, it has a higher cumulative prevalence at 82%. On 26 November 2021, the Omicron variant was classified as a variant of concern. To know more about the differences between these classifications, feel free to watch the video for SARS-CoV-2 variants on my channel. What are the key mutations acquired by the Omicron variant that makes it a variant of concern? From the genetic sequencing of many virus samples from infected individuals, it was discovered that the Omicron variant is the most mutated variant yet, which has accumulated over 49 mutations with about 30 mutations on the spike protein. This information is of great significance as the spike protein is involved in both the transmissibility of the virus and the immune response by the host against the virus. This is the general structure of the SARS-CoV-2, and the red protrusions on the surface of the virus are the spike proteins. For the SARS-CoV-2 to replicate in host cells, it must first use its receptor binding domain on the spike protein for attachment to the cells. In addition, the cell surface protease TMPRSS2 facilitates the cutting of a part on the spike protein, called the furin cleavage site, allowing the virus to enter host cells for viral replication. Moreover, most antibodies generated through infection or vaccination are stimulated by the spike protein. In other words, the immune system of the host eliminates the SARS-CoV-2 by recognizing the spike protein. From the list of mutations on the spike protein, a few mutations have been extensively studied to understand their impact. The most notable examples are the mutations that allows increased binding of the spike protein to host cells, including N501Y and D614G mutations, which are common in other variants of concern including alpha and gamma variants, Q498R, T4787K, H655Y, N679K, and mutations, with amino acid substitutions as listed. The combination of these conserved and new mutations in the Omicron variant lead to the increase in transmission rate of the virus, which led to spikes in COVID-19 Omicron variant cases that is quickly becoming the dominant strain worldwide. To know more about the impacts of N501Y and D614G that lead to increased viral transmission rate, feel free to watch the video for alpha variant on my channel. A new group of clustered mutations including N764K, N856K, Q954H, N969K and L981F mutations with amino acid substitutions as listed have been discovered in the Omicron variant. These clustered mutations reduce the efficient infection of lower airway cells that express TMPRSS2, which as mentioned earlier, is required by viral entry. Instead, the Omicron variant is capable of another form of viral entry called endosomal entry directly from cell membranes. This allowed a wider range of host cells to be infected, such as the upper airway region, leading to higher viral load and transmissibility through the upper airway. However, this is also the possible reason for lower disease severity caused by the Omicron variant, as the lower airway region such as the lungs are less affected. In addition to that, structural change of spike protein caused by mutations including G142D, Del 143 145 and Q493R leads to immune evasion of the virus, which causes a significant decrease in neutralization of virus by antibodies, as shown by the reduced efficacies of antibody treatments and vaccine immunity. In general, the mutated characteristics of the Omicron variant include potential increased transmissibility due to higher viral load. In addition, potential increased resistance towards some emergency use authorization monoclonal antibody treatments have been observed, while sotrovimab antibody treatment remains effective. 2 doses of currently available vaccines have been shown to have significantly reduced protection against infection and reinfection with the Omicron variant, when compared to Delta variant. This is due to the higher immune escaping ability of the mutated Omicron variant, combined with the waning immunity over time provided by vaccination. However, 2 doses of vaccines still lowers the severity of the disease and the rate of hospitalizations. Recently, studies show that 3 doses of vaccine protection against the Omicron variant is equivalent to 2 doses against other variants. Therefore, this provides evidence that 3 doses of these vaccines are still effective against the Omicron variant, and it is likely that this immunity requires further boosters in the future for the maintenance of the vaccine protection. On a side note, most studies carried out on vaccine efficacies use antibody levels as standard. It should be noted that most vaccines also stimulate the cellular immunity mechanisms including CD8 T cells. 80% of the binding regions of the spike protein are still recognized by CD8 T cells and are not affected by the mutations in the Omicron variant, hence giving additional protection besides antibodies. If you enjoyed this video, don't forget to like, share or subscribe to my channel. If you would like to learn more about other variants of concern, check out this series.